Alright, everyone, welcome. If you are heading into your clinical rotations, this is for you. We are going to do a really high yield review of two principles that are no doubt the absolute foundation of your practice: pharmacokinetics and pharmacodynamics. Let's jump right in. So, at the heart of all this are two sides of the same coin. You have got pharmacokinetics, or PK, which is all about what the body does to the drug. Think of it as the drug's journey. Then you have pharmacodynamics, or PD, which is the flip side, what the drug does to the body. That's its mission, its purpose, once it gets where it's going. Here's how we are gonna tackle this. First, we will look in those two core principles. Then, we will trace a drug's journey through the body using the classic acronym ADME. After that, we will zoom in on what the drug actually does at its target. And finally, and this is the key, we are gonna tie it all back to what you will be doing on the floor. Okay, section 1. Let's get this definition down cold, seriously. Getting this framework right makes everything that follows just click into place. So, what do we really mean by pharmacokinetics? Well, it's the math behind the drug's movement. It's how we study and predict and how the drugs get into the body and where is go, and how the body gets rid of it. This is all about concentration over time, which, as you know, is the difference between a drug being therapeutic and being toxic. And pharmacodynamics, this is the fun part. It's the science of the drug's action. Here, we are looking at the molecular level. How does the drug molecule bind to the receptor? What happens next? This is the mechanism that gives you the good stuff, the therapeutic effect, and of course, the side effects we always have to watch out for. Alright, on to section 2. Let's really follow a drug step by step on its journey. And to do that, we have our guiding light, that one acronym you will never forget: ADME. Absorption, distribution, metabolism, excretion. This is it. This is the entire life story of almost every single drug you will ever dispense. Knowing this sequence inside and out isn't just helpful, it's absolutely essential. So, it all starts with absorption. This is just what it sounds like: the drug moving from where you put it, say the gut for a pill, and getting into the bloodstream, into the systemic circulation. Because look, if it can't get into the blood, it can't go anywhere to do its job. It's as simple as that. And this leads us to a really big concept: bioavailability, which we just call F. If you give a drug IV, its bioavailability is a 100%. Boom! It's all in the blood. But for a pill, not so much. Most of it gets destroyed by the liver on the way. Through that's the first pass effect. So, only a fraction of the dose actually makes it into circulation. Okay, the drug made it into the blood. Now, what? Well, now it has to travel. That's distribution. This is the process of the drug leaving the bloodstream and spreading out into all the different nooks and crannies of the body. The tissues, those are in you, name it. And we have a term for this: the volume of distribution, or VD. Now, be careful. Here, this is not a real physical volume in the body. It's a theoretical number that tells us how much the drug likes to leave the blood and hang out in the tissues. If a drug has a huge VD like digoxin, it means it's everywhere but the plasma. And that's exactly why you need a loading dose for a drug like that to saturate all those tissue sites first. Another huge piece of the distribution puzzle is protein binding. See, a lot of drugs, as soon as they get in the body, they grab onto proteins like albumin. Now, here's the take home point: only the free drug, the part that's not bound, is active. The bounded drug is just sitting in reserve. It can't do anything. This is so important clinically, especially in patients who might have low protein levels. Alright, next up in the journey is metabolism. You can think of this as the body's processing plan, mostly happening in the liver. The goal here is basically to chemically change the drug to transform it into something the body can get rid of more easily. And this usually happens in two phases. Phase 1 reactions are all about modifying the drug molecule, often using that famous cytochrome P450 enzyme system. Then in phase 2, the body sticks a big water-loving molecule onto the drug, a process called conjunction. This makes the drug water soluble, basically taking it and saying, okay, you are ready for the exit. Which brings us to our final step, excretion. This is the end of the line, the irreversible removal of the drug and its metabolites from the body, and the MVP here, the kidneys. The parameter we use to measure this is clearance. Now, think of clearance as a rate of efficiency. It's not the amount of drug being removed, but the volume of blood that gets completely cleared of the drug in a certain amount of time. And as you can imagine, this is directly tied to how well a patient's kidney and liver are working. Okay, let's switch gears to section 3. So far, it's all been about the journey, the ADME process. But what happens when the drug finally arrives? This is where pharmacodynamics comes in. I love this quote because it just says it all. PK is the travel log, getting the drug to the right spot in the right amount. PD is what happens when it rings the doorbell and interacts with the target. It's the action part of the story. So the first thing a drug has to do is bind to its receptor. We measure how well it does that with a term called affinity. A drug with high affinity really wants to bind to that receptor. You can think of it as how sticky the drug is for its target. But just being sticky isn't enough. After the drug binds, it has to actually do something. And that ability is called intrinsic efficacy. It's the power of the drug to flip the switch and cause a biological response. See, a drug can have amazing affinity, but it still has zero efficacy. While we call that an antagonist, it just sits there blocking the receptor. Okay, final section. Let's land the plane here. How does all this theory actually translate into what you are going to be doing every single day as a clinical pharmacist? Here's the mental checklist you should be running through first. Always know the difference. Is this a PK issue or a PD issue? Next, have those 4 phases of ADME down cold and then know what your K parameters F, VD, clearance are telling you. Finally, you connect it all to the drug's effect by thinking about its affinity and efficacy. This right here. This is the scaffolding for your entire clinical thoughts process. So, I leave you with two clinical questions to really think about. The first one, your patient's kidneys aren't working. Well, how does the impact clearance? And what does that mean for your dosing recommendation? The second one, you have a patient with low albumin who's on a highly protein bound drug. Why is understanding that free drug fraction a life or death matter? The answers are all right here in these principles. Mastering this stuff is what makes you an expert and an absolutely vital member of the healthcare team. Thanks for joining me. If you want to stay ahead in pharmacy's new directions, subscribe to the Next Gen Pharmacist and be part of the journey.